Hello, everyone. Thank you for joining us for our third annual 24 Hours of Stem Cells virtual conference. My name is Yvonne Micah, and I support the business development of Thermo Fisher Scientific stem cell reagents and technologies. It is with great pleasure that I introduce our keynote speaker, Evangelos Kiskinis, who will be speaking to us today about the use of patient-specific iPSCs to study ALS and ion channel forms of epileptic syndromes. Evangelos joins us from Northwestern University, where he is Assistant Professor of Neurology and Physiology. Prior to his appointment earlier this year, Evangelos had earned his PhD in molecular endocrinology from Imperial College in London and completed his postdoctoral studies in the laboratory of Kevin Egan in the Department of Stem Cell and Regenerative Biology at Harvard University. It was at Harvard that Evangelos acquired his expertise using patient-specific pluripotent stem cells to investigate neurodegenerative disorders. He culminated his postdoctoral studies with a groundbreaking discovery that identified molecular pathways that become dysfunctional in the motor neurons of ALS patients, as well as the discovery of a small molecule therapeutic that is currently being tested in clinical trials for ALS patients. Evangelos has been the recipient of prestigious fellowships from the European Molecular Biology Organization, as well as from the New York Stem Cell Foundation and the Charles A. King Trust Medical Foundation. His research has also been supported by the Project ALS and Target ALS initiatives. We are very excited to have Evangelos join us today to present his newest work in his talk titled Probing Disease Mechanisms in ALS and Ion Channel Epilepsy using iPSCs, Reprogramming, and Optogenetic Approaches. Welcome, Evangelos.
Well, thank you. Thank you for that kind introduction. And I'm really excited to be part of this event. As you heard, I recently moved to Chicago from Harvard University, and I've set up my own group here at Northwestern University. And what I'd like to do over the next 35 to 40 minutes or so is tell you about our efforts to utilize iPS cells and optogenetic approaches to study ALS. And I'll focus most of my talk on ALS. I'd also like to say that the data that we'll present was largely generated while I was still a postdoctoral fellow, and I'll tell you about that work and what we're doing here to continue it. So I always like to start my talks by showing you, showing the audience a picture of stem cell-derived neuronal populations that we're using. And I do this to highlight a couple of different things. First of all, the patients that are important contributors to the research that we do. And second of all, I just want to say that this particular motor neurons that I'm showing you here, these are spinal motor neurons, and I'll tell you a little bit about how we make these. Belong to an ALS patient that was diagnosed with a disease a few years ago. And they actually passed away a little short after they donated a little piece of their skin to our lab. That was more than 8 years ago. And it has only been through the transformative work of iPS cells and reprogramming that we've been able to study these patients, study their motor neurons, and try to get to the bottom of why, what was wrong with them and why they discovered the pathophysiological mechanisms of ALS. So I'll give you a little bit of a brief introduction on ALS. ALS is, stands for amyotrophic lateral sclerosis. In the US is also known as Lou Gehrig's disease, and through the world right now it's also known as the Ice Bucket Challenge disease. So it is a devastating neurodegenerative disease that is caused by the specific loss of a very specialized neuronal subtype called the motor neuron. Now there are two types of motor neurons, the upper motor neurons that are found in the brain, and then the lower motor neurons that are found in the spinal cord. And on the right part of the slide, there's an illustration of an oversimplified illustration of the motor neuron circuit. You can see the upper motor neurons synapsing with the lower motor neurons, and the lower motor neurons send their axons outside the central nervous system to connect with muscle through the formation of a very specialized synapse called the neuromuscular junction. And they control movement. They control all types of movement, from how we speak to how we talk to how we breathe, how we walk. And when a patient is diagnosed with ALS, they gradually lose the ability to control their muscles because these motor neurons degenerate. Now, the disease clinically is characterized by spreading, which leads to an eventual fatal paralysis. The age of onset is about 45 years, and it's a very aggressive disease. So the average survival ranges between 2 to 5 years from diagnosis to death. Now, it is a disease that has variable clinical presentation. So some patients present themselves with an ability to move their index finger. Other patients present with an inability to move a limb, or a whole limb, or an inability to talk. And we're not entirely sure what is behind this variable clinical presentation, particularly because in many instances, patients that have the same genetic mutation show this clinical variability. So the other thing that's intriguing about ALS is it's genetic. So there's been tremendous breakthroughs by a number of initiatives and genetic researchers over the last few years. And we now have identified Mendelian variants in more than 20 genes. In addition to that, there's multiple candidate susceptibility genes that contribute to ALS. Now, what is intriguing about this is that these genes code for proteins that seemingly do very, very different things. Now, the first gene that was ever discovered to cause ALS by Bob Brown's group at MGH was SOD1, highlighted, shown here on the right, which stands for superoxide dismutase. And I'll tell you a little bit about this, or a lot about this gene today. More recently, a gene which codes for a protein with an unknown function called C9orf72 is by far the biggest genetic contributor of ALS. And there's other important players such as TDP43 and FUS. And I'll tell you a little bit about these different genes. Now, importantly, the overwhelming majority of ALS is still of a sporadic nature. So we estimate that familial cases account for 10 to 12%, and the rest is of a sporadic nature. And one central question that's still outstanding in the field is, how are these different genetic subtypes of ALS related to each other, particularly in terms of the molecular mechanisms that lead to the degeneration of the motor neurons? And how do these genetic subtypes relate to the to the majority of sporadic cases that are out there? This is a very important question. It is a question that we are interested in addressing using cellular models of disease. Now, the other thing that has complicated studying ALS is the fact that multiple cell types are implicated in pathological mechanisms. Now, we know this through work that has been done in multiple labs over the last 15, 20 years. And while it is clear that it is the motor neuron that's, at least in the initial stages of the disease, targeted for selective degeneration, we know from in vitro and in vivo work that other cell types, such as astrocytes and oligodendrocytes and microglia, which are found in abundance in the central nervous system, are critical, particularly for the progression of the disease, but probably also for the onset of the disease. Just to summarize this first part, and the reason why I want to do this is because I want to highlight how iPSCs and reprogramming approaches could be useful. So I told you that in studying ALS, I told you that the disease is clinically and genetically very heterogeneous. It involves complex cell-cell interactions that are hard to decipher in vivo. It is not possible to isolate motor neurons from living ALS patients. And obviously, we can't create any animal models of a sporadic disease. Now, last but not least, it is obvious that by the time of diagnosis, the disease processes are well underway. So we, and obviously many others, have suggested that pluripotent stem cells and generally reprogramming approaches could provide a potential solution to some of these issues. Now, I'm sure that most of you have seen one version of this schematic in one form or another, but this nicely summarizes the approach that we take in my lab. So we identify a patient that we want to study. We isolate some sort of somatic cell type from that patient. This can be skin fibroblasts or it can be blood cells. We reprogram these somatic cells to generate patient-specific iPS cells. Through the use of genome editing approaches, we either repair the mutation in cases where we know what the mutation is, or we will take a wild-type iPS line and introduce a genetic variant in case we want to test the effect of that genetic variant. Now, this is a tremendous tool because it allows us to generate isogenic stem cell lines, which are a great control for when you're doing this in vitro modeling experiments. And once we have these cells, we in vitro differentiate them into different neuronal subtypes. In the case of ALS, these subtypes would be spinal motor neurons, but we also make different cortical subtypes such as excitatory glutamatergic or inhibitory GABAergic interneurons. That's to study epilepsy. And once we have these cells, we compare them through a combination of classic molecular biology approaches and genomic analysis, such as global expression analysis, functional and biochemical experiments. And the goal here is to identify pathways, identify specific targets that we can go after using small molecules or other approaches. And hopefully I'll illustrate how we've gone about this cycle by focusing on a couple of ALS patients. So the first obvious question that we wanted to address when we started this work was, well, if we generate patient-specific iPS cells from ALS patients and we generate their motor neurons, do we see phenotypes that could be related to the disease? So to address this question, we initially focused on two patients. Both of these patients were female, diagnosed with the same genetic variant that was a A4V mutation in the SOD1 gene. This is the most prevalent mutation in North America, and it's a very aggressive type of genetic ALS. Both of these women had a relatively young age of onset of 43 and 45 years of age, and both of them actually passed away within 2 years after the initial diagnosis. We made the iPS cells, and we initially decided to compare the motor neurons that we're generating from these patients to two age-matched controls. How do we make the motor neurons? So we've adapted protocols. This is largely work based on Nick Witchley's work at Tom Jessel's lab and work done in Laurent Studer's group. But we've adapted these published protocols, and we've come up with a motor neuron differentiation protocol, which lasts about 24 days. So it takes us 24 days to make the motor neurons, to make post-mitotic motor neurons, I should say, from the stem cells. At the end of 24 days, we replate these motor neurons, and then we grow them for anywhere between 5 to 30 days. And at various time points that I will illustrate throughout the talk, we study these motor neurons. Now, there are two important steps in this protocol. The first one is the dual SMAD inhibition, which forces all the stem cells down the neuroectodermal lineage. And the second step is achieved through the addition of retinoic acid and a small molecule agonist of the sonic Heterogeneous pathway, which patterns these neural progenitors to form spinal motor neurons. Now, our group, Kevin's group, and a number of other groups have spent a lot of time trying to demonstrate that these motor neurons that we make are real functional motor neurons. So we know that these, and I'm summarizing in one slide, years of work essentially done by multiple groups, but we know that these motor neurons express the right transcription factors. I'm showing you here islet and HB9. These are motor neuron transcription factors. The majority of them are FOXP1 positive, which would indicate that they belong to the lateral motor column. And of course, over time, they express CHAD, which is the enzyme choline acetyltransferase that makes acetylcholine, which is the primary neurotransmitter of motor neurons. We know that these are electrophysiologically active. They fire spontaneous and induced action potentials. They're able to respond to excitatory and inhibitory signals. And in collaboration with other groups, we've shown that if we inject purified motor neurons into developing chick embryos, they will functionally integrate in the ventral horn, and they're able to form neuromuscular junctions. So one of the first things that we did when we made these motor neurons from the two patients, these patients are known as 39B and RB9D. That's just the nomenclature that we used in the lab. So these patients are de-identified. And we compared how these motor neurons survived over time in culture relative to two age-matched controls. We noticed that in the cases of the two patients, the motor neurons survived, or the numbers of motor neurons after 30 days in culture were significantly lower than those of the controls. Now we know that these differences are not due to differential proliferation, and we think that they're due to selective apoptosis, because we've done BRDU incorporation assays and tunnel assays and so on. Importantly, we also know that these differences are specific to islet TJ1 positive motor neurons, whereas islet negative generic neurons in those cultures do not show these same changes in numbers. We think that these are specific to motor neurons. Now, of course, we and others are very mindful of the fact that there's incredible genetic and epigenetic variability in different IPS lines, and this variability can confound conclusions regarding identified phenotypic differences. And you can have instances where you're looking at a phenotype such as survival, for example, or morphology, and this phenotype can be due to variability rather than the disease itself. Now, there are many approaches that help you overcome this problem, but probably the most the best way to overcome it is through employing gene targeting technologies to actually repair or introduce the genetic variant to test whether the genetic variant is necessary or essential to introduce these phenotypes. So we decided to do that, so we chose one of the two IPS lines. I'm showing you here at the top a sequencing graph illustrating how the patient-specific IPS line from that original patient has heterozygous mutation in the SOD1 gene. This is in the first exon of the gene. And through zinc finger-mediated gene targeting, we were able to generate an isogenic control stem cell line in which we had fixed, we had repaired this mutation. Now we know that through the repairing process, we did not mess up the genome. We actually did a whole genome sequencing analysis to show that there were no differences between these two lines other than the presence of the disease-causing mutation. So now that we had this isogenic control, we went back to our culture system and we asked, where can we rescue some of these phenotypes that we originally identified? And on the left, I'm showing you the motor neuron numbers, again, after 30 days in culture, and I hope you can appreciate that. On the far left, you're seeing the numbers of the patient line, the one which has the heterozygous A4V mutation, and next to it, you're seeing the isogenic control, and I hope you can appreciate that the motor neuron numbers have significantly increased when we fix the mutation. We're also able to show that the average size of the soma of these motor neurons also increases as we fixed the mutation, we had originally shown that there was a difference between patients and controls. Now that we had phenotypes that we felt confident about, the important question is, well, can we utilize this system to identify what's behind these phenotypes? Can we identify molecular pathways that mediate these differences? Now, to address this question, we decided to take initially an unbiased approach, and we did an RNA-Seq experiment. Now, the way we do this is, as I mentioned before, it takes us 24 days to make the motor neurons. At the end of those 24 days, we plate motor neurons in a co-culture system with mouse glial cultures. We think that this is crucial and it's important to mature these cells. But then to purify the motor neurons out of this culture, we utilize a lentiviral plasmid, which expresses a reporter, RFP, under the control of a motor neuron-specific promoter, that is the HB9 promoter. So we infect our cultures with this HB9 RFP virus. And 15 days later, we fax purify our motor neurons, and we did an RNA-Seq experiment. Now, we chose to look at these cells on day 15 because we knew that at that time point there are not statistically significant phenotypic differences, at least in terms of the survival and the morphological aspects of these motor neurons. So on the left of this slide, I'm showing you a classic cluster dendrogram, which illustrates that the parental mutant patient motor neurons, illustrated by the two branches on the right side, nicely separated from the isogenic control corrected. And we analyzed the significant differences between the isogenic motor neurons by a number of different ways. The gene set enrichment pathway analysis was highly informative, and we were excited to see that pathways like mitochondria, oxidative phosphorylation, and electron transport came up as being significantly enriched in being dysregulated. Other things included protein translation, protein folding, transport proteins such as kinesins and dyneins, and very importantly, a number of ion channels came out of this analysis. So next, we tried to basically validate this gene expression differences by doing a number of functional assays. So the first thing that I want to show you is what we did is we found that if you look at genes that code for proteins that are involved in protein translation, we identified 139 of these proteins as being expressed in the two different types of motor neurons. We found that the overwhelming majority of these, so 130 of 139, were significantly downregulated in the presence of the mutation. So we thought that this was particularly intriguing because downregulation of translation is a hallmark of a very well-characterized pathway known as ER stress and an unfolded protein response. Now, what is this unfolded protein response? Well, this is a natural self-defense mechanism that cells utilize to protect themselves from various stress insults, such as the presence of misfolded proteins. And we knew from previous work that mutant SOD1 and SOD1A for V mutant protein is misfolded. That was work that was done in mouse models of the disease. So what the cell does when it detects misfolded proteins is it basically says that I need to shut down translation until we deal with this misfolded protein. Now, in order to deal with this misfolded protein, what the cell does is upregulate the expression of specific chaperones and other foldases that will help refold these misfolded proteins. But importantly, if the cell cannot deal effectively with the burden of misfolded proteins, then this pathway can become apoptotic. So initially itself it's protective, but over time it becomes apoptotic. In order to validate that this pathway was indeed induced in our cells, we decided to look at two hallmark markers. The first one is the phosphorylation levels of this protein called EIF2 alpha, which actually mediates the downregulation of translation. and the other one was the splicing of a transcription factor known as XBB1, which in response to this misfolded protein gets spliced and mediates downstream effects. So what I'm showing you here is a Western blot that was done in mixed motor neuron cultures at different stages after the differentiation from the mutant and the isogenic control corrected on day 7, day 9, and day 12. And on the top, I'm showing you the phosphorylated levels of AF2 alpha, and at the bottom, I'm showing you the total levels of AF2 alpha and the normalization. And I hope you can appreciate that indeed we found evidence of increased phospho-AF2 alpha at all the different stages of the motor neuron cultures, indicating that this pathway was indeed induced in our cultures. Now, when we looked at the splicing levels of XBB1 itself, we also found evidence that for increased XBB1 splicing, which was decreased when we fixed the mutation. Now, obviously, these differences are not dramatic, but they're reproducible and statistically significant. So now that we have these markers, well, we said, how important are they for this phenotypic difference in terms of the survival of the motor neurons? So we went back to our cell culture system, and we decided to use an siRNA approach to knock out either XBB1 or ATF4, which is downstream of phospho-AF2 alpha. And what we found was that if you knock down XBB1, you see a small but nevertheless significant increase in the survival of specifically the mutant motor neurons, whereas if you knock down XBB1 in the isogenic control motor neurons, that, if anything, has a negative effect, which indicates that motor neurons require some levels of spliced XBB1 just to stay alive. Whereas if you knock out ATF4, this had a detrimental effect on the survival of both mutants and controls. So our interpretation of this data is that perhaps splicing XBB1 is important for this degenerative phenotype that we've identified, but it's not it's not the only factor that contributes to it. Whereas ATF4 seems to be essential for the survival of these motor neurons, at least in vitro. So I also mentioned that we identified a number of ion channels that were significantly misregulated in our culture system. Now, in order to address any electrophysiological alterations that would happen in response to these ion channel differences, we decided to turn to a novel approach known as the Optopatch. This is a method which was developed in Adam Cohen's lab at Harvard University, and Joel Kragi and Daniel Hochbaum, two brilliant fellows in Adam's lab, were crucial in developing this technology. Now, this is a very clever way of doing electrophysiology at a large-scale format, and it's dependent on the co-combinatorial expression of two very specific proteins. First one, and I'm showing you here on the right, is a classic channel rhodopsin. So this is a protein that, upon blue light stimulation, triggers an actual T cell triggers neural activity. And the other protein is called an archrhodopsin, and the archrhodopsin basically does, in a way, the exact opposite of what the classic channel rhodopsin does. It emits light in response to a change in voltage. So optogenetic actuation occurs through this blue light activated channel rhodopsin, which is called sheriff, and the voltage imaging occurs through a near infrared genetically encoded fluorescent voltage indicator known as quasar. So we decided to basically co-express these two proteins in our motor neurons and utilize them to record activity. Now, Adam Cohen's group has developed specialized optics and software which allows for simultaneous simulation with the channel rhodopsin and recording from large numbers of single cells. And I just want to highlight a couple of figures from the original publications. This is one figure from Hochbaum et al., which published at Nature Methods. And what I'm showing you here is a cell that co-expresses the channel rhodopsin and the quasar. Now these constructs have reporters so we can visualize them. And what Adam's group did here is they recorded the optical activity, the optical signal, but at the same time, this cell was passed. And what I'm showing you here at the bottom is the optical reading. So those are the red lines. And in the dark lines, you can see the actual voltage reading as read by the patch. And I hope you can appreciate that the recordings, the traces closely correspond. Now, importantly, its blue dot here indicates a blue light stimulation. So you can see that when we stimulate these cells with blue light, you get a triggering of an action potential. So this technology is incredible, and here I'm just showing you a movie again from the original publication showing actual spontaneous network activity in a co-culture of primary neurons with glia. And for the first time, somebody can visualize real voltage in neuronal culture. So we utilize this technology. We infected our SOD1A4V and our isogenic control corrected motor neurons. And the strength of this is that it's incredibly high throughput. So literally within a couple of experiments, we're able to record from about 600 cells. And what we're able to do with this is we can record spontaneous activity, but we can also record activity in response to different amounts of blue light stimulation. What I'm showing you here in this graph is how we employed 10 different steps of blue light stimulation, whereas with every step we increased the amount of blue light, and therefore we increased the amount of voltage. And I hope you can appreciate that the mutant motor neurons, indicated by the red lines, seem to fire increasingly more in between the blue light stimulation, so in between each step. And they seem to fire more only through the first couple of steps of blue light stimulation, whereas in the later steps, so as we increase the blue light stimulation, the mutant motor neurons tend to fire less. So here we have a reversal of the phenotypes. So under conditions of low stimulation or under conditions of no stimulation, we see higher spontaneous firing, higher firing rate in the mutant cells, whereas under conditions of high blue light stimulation, we see decreased firing rates of the mutants. On this slide, I'm just showing you a quantification of these processes. This is the spontaneous firing rate. This is recordings down from about 1,000 cells from the mutant and the isogenic control motor neurons, and you can see that the mutants fire spontaneously more. And here I'm showing you the number of action potentials that the mutant and the control motor neurons fire in response to different intensity stimulus. So these are the different steps. And you can see that at the lowest levels of stimulation, the mutant motor neurons fire significantly more, but as we increase the stimulation, the mutant motor neurons fire significantly less. So to get to the bottom of this interesting phenotypes, we reverted back to doing classic PATSCOM recordings. This is in collaboration with Brian Wanger and Clifford Wilt's group. And again, we utilize the HB9 lentiviral approach to identify specific motor neurons. And what Brian did was he was able to show that in the mutant and the isogenic control motor neurons, found that these excitability differences arise from a reduction in primarily potassium currents. So now that we had a number of pathways, so excitability and ER stress, and we had a specific target, potassium currents, we decided to test the hypothesis that if we target these pathways through small molecules or drugs, maybe we can see if they would have an effect on the cells. So we tried a number of small molecules, and I'm just showing you here a drug called retigabin. Retigabin is an FDA-approved drug that is currently used for the treatment of epilepsy And what we found when we applied this drug on our mutant and uncontrolled motor neurons was that it was very effective. Retigabine works by targeting a specific subtype of potassium currents known as Kv7 channels. And we're able to show that if we treat our motor neurons with retigabine, we're able to restore normal physiological activity in these cells. We went back to the survival paradigm, and we treated a number of control lines and a number of, or our 2 patient lines, in this case with retigabine, for a number of days. So this is 2 weeks treatment with 10 micromolar of retigabine. And what we discovered was that retigabine was very good at increasing the survival, particularly in the case of the mutant motor neurons. So now that we had a target, well, an important outstanding question was that, well, we know that there's a mutation in SOD1. So how does mutant SOD1 relate to these different molecular pathways that we identified as being misregulated in our cells? Are these indeed mediated by misfolded SOD1? It's important to say that SOD1 patients in postmortem biopsies show aggregations of misfolded SOD1 in their spinal cords. And an important and outstanding question is whether these aggregations correlate with pre-survival, or are they in any sort of or sense protective. So we went back to our culture system and we decided to look for visible SOD1 aggregates. And we tried a number of different antibodies in a number of different ways, and we do not see any microscopically, at least, visible SOD1 protein aggregates accumulating in our motor neurons under these basal conditions. Now, it's important to state that it is at the same time under these basal conditions that we see all these other phenotypes happening in our cells. Prove to ourselves that we could indeed detect SOD1 aggregates, we decided to do a control experiment in which we treated our motor neuron cultures with MG132, which is a proteasome inhibitor. And when we did a short-term treatment at 1 micromolar with this inhibitor, we were indeed able to identify microscopic aggregates of SOD1, which happened at a significantly higher rate in the mutant motor neurons and not in the isogenic controls. We think that there is misfolded SOD1 protein. And another way we looked at this was through a classic biochemical approach. So this is doing a Western blot and looking at the soluble versus the insoluble fraction, again under basal conditions and under conditions where we treated our cultures with MG132. So if you concentrate on the conditions without MG132, so these are the columns indicated, the second and the fourth column on the slide, I hope you can appreciate that under the urea soluble conditions, there's no SOD1 detected, but under conditions where we stress the cells by blocking the proteasome specifically and detect SOD1 only in the case of the mutant motor neurons in the insoluble fraction. So what is this telling us? This is telling us that under normal basal conditions, there is no aggregated SOD1 and there is no insoluble SOD1. But if you block the proteasome, then you see a specific and selective upregulation of aggregation and misfolded and insoluble SOD1 only in the case of the mutation. Now, to look at this further, we collaborate with Jonathan Gilthorpe's group, who has developed a very elegant ELISA assay, which allows to detect misfolded but soluble SOD1. And in collaboration with Jonathan, we're able to show that in the presence of the mutation, we see accumulation of misfolded but soluble SOD1 protein, specifically in the case of the mutant motor neurons. We think what is happening is that there is, in the present mutation, there is misfolded SOD1, but this misfolded SOD1, at least under these basal conditions, and at this time point, it's soluble, and it is the soluble form of SOD1 that causes these downstream problems. Now, another important question that we wanted to address is how are these two different pathways connected, if at all connected, in these ALS motor neurons? So one important observation that we made early on through our experiments was that if you look at the levels of XBB1 splicing as a marker of ER stress in motor neurons versus a range of other cell types that we can generate in our lab, we identified that purified motor neurons seem to have higher levels of XBB1 splicing, which increase over time in culture. So I'm showing you here XBB1 splicing levels in purified motor neurons over time. On the far right, I'm showing you positive control. These are fibroblasts treated with DDT, which induces ER stress. And you see that in that case, most of the XBB1 is spliced. And I'm showing you the levels of XBB1 splicing in a range of other cell types, including non-motor neurons that we made from embryonic stem cells, ES cells, neural progenitors, astrocytes, and fibroblasts. And I hope you can appreciate that wild-type healthy motor neurons have higher levels of basal XBB1 splicing. Now, we think that this is not an in vitro artifact because if we look at RNA isolated from human spinal cord and human brain, we indeed identify significant levels of XBB1 splicing, specifically in the spinal cord and not in the brain. So we were wondering how these excitability defects connect with the induction of ER stress in UPR, if at all. To address this question, we decided to do a simple experiment, which was to manipulate either excitability and read ER stress levels, or manipulate ER stress and read excitability levels. And we did this through the use of small molecules. I'm not going to show you all the experiments, but what I'm going to show you is two experiments that we did. So on the left, I'm showing you that when we treat motor neurons with salubrinol, which is a molecule that's been well documented to reduce the levels of ER stress, that leads to a reduction in the number of action potentials that are being fired. Whereas if we increase firing by treating our motor neurons with linopyridine, which is a Kv7 antagonist, or kainate, we increase XBB1 splicing as a marker of ER stress. So in other words, if you manipulate ER stress, you affect excitability, and if you manipulate excitability, you affect ER stress. So our working model is that there seems to be a connection between the levels of ER stress and excitability. This is we're looking into how this connection is mediated. We suspect it's through calcium, but we haven't proven that yet. We think that this connection is something that happens in generic spinal motor neurons, but when you have an insult such as the presence of a mutation in SOD1, which leads to further ER stress, this the stress feeds into the cycle and leads to a vicious cycle of excitability and stress, which leads to the effects and the degeneration of the motor neurons. Now, importantly, everything that I've told you about today is focused on SOD1 cases, and I've told you that SOD1 accounts for a very small proportion of all ALS cases. Now, the strength of iPSCs modeling is that you can address how important your findings might be in other subtypes. So we decided, such as CNRF or FUS, other genetic subtypes of ALS. So we decided to address whether these different genetic types of ALS converge on these pathways. So to do this, we decided to focus on excitability, and we used a multi-electrode array system. This is the Axion biosystem, which allows for recordings of spontaneous physiological activity from populations of motor neurons. The strength of this is that you can grow your motor neurons in these plates, which have electrodes at the bottom of these dishes. These are shown here. and every once in a while you can take these plates, load them on this machine, and record spontaneous activity. Here I'm just showing you a movie in which you're looking at visual representations of the activity. Each column represents different replicates of either a patient and an isogenic control. And I hope you can appreciate that in the case of the patients, although there's variability, there's significantly more spontaneous activity, which is reduced when we fix the mutation. So we decided to utilize this technology and make motor neurons from a high number of patients and controls. This was a heroic experiment. It took 4 people working full-time for 3 months to do this experiment because we had to, at the same time, differentiate motor neurons from 20 different lines. We plated these motor neurons on these multi-lecture array systems, and we measured spontaneous activity. So this graph illustrates the average spontaneous activity in a number of control individuals, in a number of SOD1 patients, CNRF patients, and FUS patients. And I hope you can appreciate while there's high variability even amongst the controls, as a group, the SOD1 cases, the CNRF cases, and the FUS cases, those are different genetic subtypes of ALS, significantly segregate from the control population and showing higher spontaneous activity. So this experiment was important because it illustrated to us that our findings with the SOD1 patients were perhaps broadly applicable to other genetic subtypes of ALS. So what I've shown you is basically how we've started from from 2 ALS patients. We generated patient-specific iPS cells from those patients. We utilized genome editing approaches to generate isogenic controls. Differentiated motor neurons from these pairs of isogenic lines. Studied these motor neurons. We identified a series of molecular pathways such as the stress and excitability dysfunction. We identified pathways and targets such as potassium currents. We identified small molecules and a particular drug which targets a pathway. We illustrated that this drug can increase the survival and can effectively restore excitability in these motor neurons. And we also showed you that these motor neurons, that these effects are broadly relevant to a number of genetic subtypes. Now, based on this work, there's currently an ongoing clinical trial. This is a phase 2 pharmacodynamic trial for this small molecule, for this drug, reticabine, for ALS patients. This clinical trial is run by my great collaborator and former colleague Brian Wanger at MGH, and a number of funding agencies are involved in this, including GSK, MGH, HSCI, ALSA, Target ALS. And what's particularly fascinating about this trial is that it's going to look at 120 different patients, and we're going to make 120 different iPS lines from each individual patient to look at the effects of these drugs in vitro, and in vivo. So we're particularly excited about where this is going. I just want to finish by saying that what we are trying to do is we're utilizing the same approach to try and decipher what is the molecular taxonomy of the disease. So we're trying to determine how similar or different genetic and importantly sporadic subtypes of ALS in terms of the molecular mechanisms that are targeted in the motor neurons for degeneration. We think that this is important because if we identify common or unique pathways that are affected, this will help us design better clinical trials and hopefully lead to personalized therapeutic approaches. Now, I want to thank the people and the funding agents that were involved in this work. So for all the optogenetic approach, Joel Craggy, Peng Zhao, Eli Weinstein, Adam Cohen, and Daniel Hochbaum, for Brian Wager and Clifford Wolfs for the electrophysiology, Gabriella Bolting, Jack Sando, and Luis Williams in Kevin's lab for various aspects of the iPS disease modeling. I'd like to thank my own lab, which is taking this forward taking this work forward and various finding agencies. So that's where I'd like to stop. So thank you for your attention. I'll be happy to take questions.
All right. Thank you, Evangelos, for that very interesting presentation. We now have time for a live question and answer session. So for those of you who are joining us live, you can use the chat box in the lower right corner of the window to send in your questions to Evangelos. So this is a great opportunity for you to connect with Evangelos and ask him all your burning questions. So go ahead and send those in. We've already had a few come in during the presentation, so we'll get started with those. The first is, does expression of the two bacterial rhodopsin molecules modify the natural electrical response patterns of the neuron?
Yeah, thank you for that. That's a great question. So the short answer to that question is no. So we've done a series of very careful experiments where we've used classic PATS physiology to look at a number of physiological properties of motor neurons that express this channel versus motor neurons that do not. And what we have found is that the physiological properties of these cells are not affected when they express these channels. So we have a study that's currently in review that will summarize all the optogenetic approach, and those control experiments are included in that study.
Fantastic. We're getting a couple of questions about aging. So are you thinking about incorporating mechanisms for aging into your disease model?
Yeah, so that's a critical and very important question, particularly because, as I mentioned at the beginning, ALS is an adult onset degenerative disease. So a question I always get, and a question that I think about a lot, is how come you are seeing these phenotypes in the first place in motor neurons that are young and only after a short time in culture. So I like to think about this in a certain way. So I think that there are three different phases in the experiments that we do. The first phase includes the developmental phase. So, and I think that is how you go from a human embryonic stem cell or an iPS cell to a post-mitotic neuronal subtype. Then there's the second phase, which I like to think as a maturation phase. So how do you go from a post-mitotic motor neuron to a mature functional motor neuron? And the third phase is how you go from a young motor neuron to an old motor neuron. And I think we've as a field we've done well in deciphering the development and the maturation phase through a number of different approaches. But aging remains challenging. There's been certain breakthroughs. A highlight of which is the work done from Lawrence Studer's group, where they artificially age their neurons by expressing progerin. And while that is one approach, I think there are a number of different approaches in the works that, and I believe that we're going to see breakthroughs in the near future. And we are actively engaging on figuring out how to age our motor neurons in cultures, and hopefully I'll be able to talk to you about this next time. I think what I have to say is I think an important question is, at least in the motor neuron field, is what is lacking is knowledge about how a young versus an old motor neuron differ. And without that knowledge, and that's in an organism, like in a human or in a mouse, and then without that knowledge, it's hard to devise methods of how to age our motor neurons. Now, underlying this question is this idea of why are you seeing these phenotypes when this is an adult onset disease? And again, this is a question I think about a lot. There's many potential explanations. An in vitro environment is stressful in itself. These cells do not exist in isolation in people. So my view is that has a lot to do with it. Another important thing to consider is while a number of these degenerative diseases are indeed adult onset and it takes years for them to be clinically obvious and diagnosed. I would argue that we do not know how a 6-month-old patient individual that's born with an SOD1 mutation looks like, and particularly how their motor neurons look like. Yeah, I hope I've addressed that question effectively.
I think those are great points, and we're certainly looking forward to learning more about that in your future talks. You mentioned the microenvironment and that these cells don't exist in isolation. We had a question come in from the UK asking, are there additional insights to be gained from co-culturing your cells with human glial cells, perhaps iPS-derived glial cells?
Yeah, absolutely. That's a great question again. I think this is an important and outstanding thing that we need to do. It makes perfect sense. If you think about it, this, in an individual that has this mutation, it is not only their motor neurons that have the mutation, it is their astrocytes, their oligodendrocytes, their microglia. And I think the answer to the question is yes. I think that there'll be important insights to gain from that. I think it has been particularly challenging to derive iPS-derived functional astrocytes. I think there's been breakthroughs in that regard recently with direct conversion methods from the Broccoli group. And I think that it's important to do these experiments. I think as a field we need to move towards more sophisticated cell culture models where we recreate as well as we can the central nervous system with connections and the different subtypes that exist there, as well as the three-dimensionality of it. And I think as a field, we're moving towards that direction. So absolutely, I think these experiments need to be done. We have not done these experiments particularly because we find that the astrocyte protocols are very challenging.
Fair enough. Well, we'll look forward to hearing more about that as well. And I think we have time for one last question. Will there be a difference in exosome content between normal and ALS motor neurons?
Yeah, that's a great question. Unfortunately, I do not know the answer to that question. We haven't looked at that at all. But I'm aware of studies done in other degenerative diseases that have looked at this. So I think it's an important thing to do. It will be particularly interesting to look to see whether misfolded SOD1 is released through exosomes and amongst other things. So I think it's a great idea.
All right. Well, I know we've come up on the hour in the conclusion of this talk, so I want to take this moment to thank you, Evangelos, again for joining us today and sharing your exciting new story. And I also want to thank everyone in the audience who signed on today to join us for this talk. For those of you who are interested in learning more, we do have a live chat that will be happening in the stem cell research booth in the exhibit hall. The scheduled live chat is hosted by 2 of our R&D experts and will focus on approaches and considerations we face for neural disease modeling. So go ahead and pop on in to see that conversation and ask our experts your questions. So with that, I'm going to go ahead and conclude this session, and thank you again, Evangelos, for sharing your very exciting research with us today.
Yeah, absolutely. Thank you, and feel free to get in touch with me through email if you want to discuss or continue the conversation. Thank you so much for your time.